are basically annotated by functional and by BLASTX. After that, we'll try to find some expression analysis of particular genes by using BOTI2, which is basically for mapping the raw reads back to the assembled sequences. After that, we'll calculate the RPKM values of particular reads from disease and normal conditions, and then we'll draw a particular heat map of the data. But unfortunately, I'll not be able to do BOTI2 in my laptop, but I'll show you what is the command line. In previous lecture, I have shown you what is the command line for filtering, and for this particular BOTI2, I'll show you what is the command line and what are the basically steps that you have to perform in Linux system only. Okay, for Windows system, it will not work because our I don't have so much memory in my system right now. But whenever I'll do work with my workstation, definitely I'll share that tutorial with you. Okay, but currently I'll show you only the results related to AgriGeo. Okay, I'll I'll share my screen. Let me find some sequences and then I'll put those sequences in AgriGeo, and then I'll show you what type of functions are there, and I'll share my presentation with you as well. My screen is visible.
No, sir.
Yeah, screen is visible or not?
Yes, it is.
Okay. So, for example, this is the AgriGeo tool that I have opened for particular plants only. Currently, I'll show you the result of some plants. So I have previously shown you how to download the data, how to assemble the data, but currently I don't have any assembled data. If you have any assembled data, have you assembled the data in your own laptops that I have shown you the technique by CLC Workbench?
No, sir, because I don't have the tool access.
Okay, no problem. So what I'll do, I'll just download the data and I'll try to find some annotations of the data. Okay. For example, at NCBI. So what I'll do, I'll select these 20 sequences just for trial. And then I've downloaded these sequences. My screen is visible. Hello, I'm audible.
The screen is not visible now.
1 minute, 1 minute. Is it visible? Okay.
Yes.
So what I'll do, I'll compare my data. First of all, this is based on Arabidopsis thaliana only. Okay, means I have to select some model organism related to, for example, Arabidopsis, because I have selected T only. T is not available. Camellia sinensis, that is not available in this particular chart for ontology. Today we are doing ontology of a particular organism. Genome is available, but ontology is not available right now for this particular plant. So what I have to do, I have to find which is the closely related plant which is nearby this Camellia sinensis. The genome of that particular plant is overall explode. So what we have to do, we'll go towards tear. As tear is available in this particular community, Camellia sinensis is not available, and tear is fully annotated. Camellia sinensis is still lacking here. So what we have to do, we'll select some model organism, that is tear. And what we'll do, we'll blast our sequences with tear. This is a tool. There is a blast in tear as well. We'll put our sequences here that we have selected. For example, this Camellia sinensis, first, first, I have to select this sequence and try to find what is the pair ID of this particular sequence. So I'll run the blast and try to find the particular ID of this sequence in TER database. So this is the result. I have found the top hit as 83G like this. Okay. So I'll select this in this. Again, for second sequence, I'll repeat this thing, repeat, and again, maybe I'll find some for 100 sequences, I may be able to find some 10 tier ID, 50 tier IDs or 60 tier IDs, and I'll copy all those tier IDs into this particular agrigio, and then I'll try to find some particular annotations at cellular, biological, and molecular levels. So by selecting these 20 sequences, what I'll do, I'll just copy the whole data and then I'll run blast. But before that, I'll need, I don't need 50 alignments. I need only single alignment. Okay. And then run blast. Here sequence length is high, more than 40,000. So it will not provide me results. So I have to reduce. Either this is the one thing. And second thing, what we can do, we'll go towards download and go towards this particular protein section, and we'll download whole list of these proteins. And by using remote blast, we'll be able to find which is the related sequence with our targeted organism. So I'll extract once we download it. But in case of humans, genome is already available. You don't need to find some model organism. For example, my first sequence is related to this particular sequence, second is related to this, third is related to this, for example. Okay, so I'll select randomly 20 sequences. More than that, First of all, I have to organize those genes. All we need to separate this. Now easily I can copy all these pair IDs. Will not recognize our faster sign. So we have to remove this. These are the IDs, for example, we got after our tear blast. And I'll put all these IDs here. And I'll submit my data to. That only 10 entries are mapping. That was tier 10. I have downloaded data for tier 11. So I've selected the whole data. Now, this is the result that I got from these tier IDs. First of all, I'll download this result. That what I have. That from this particular tier ID, this is the gene ontology. Now, what is this gene ontology? How we can find what is the basically result of this gene ontology? So what will we do? We'll see the biological processes, cellular components, and molecular functions. First of all, we'll generate some details regarding biological processes. We can generate image as well. We can download the details from this as well. First of all, we'll generate image. Now see, in this particular biological process, first of all, what these colors are representing. If arrow is like this, This is a part of, this is a positive regulator, this only regulates, this is a negative regulator, there may be two significant loads, and this is the levels, means how much this is regulating, up regulating or down regulating, whatever it is doing. So we can easily find what is our target. Might be we are targeting some reproductive processes, might be we are targeting some developmental processes, might be we are targeting some other cellular processes or some metabolic processes. We can easily find our data what based on our target only. So this is the particular biological process. But if we need in more concise way, then there is a more tool known as Vigo. It will directly analyze the results. First of all, we have to download these results. Download it. Now, what I have to do, I'll input my results in Vigo format. Now, Vigo has given us this type of results. Now, see geoterms regarding biological, cellular, or molecular functions. So, how to find these cellular, biological, or molecular functions? We have to see this is the cellular component. We'll select molecular functions and then biological processes. These are all IDs. For example, in biological processes, this is response to stimulus. This is for molecular function. We can easily find the list of genes as well, then cellular components. By this way, we can easily find the summary of biological, cellular, biological processes, cellular components, and molecular functions. So, why I'm doing this, I'll share my presentation with you, either my publication with you, where I have done this work. I'll share publication. I think that would be better. I'll share publication related to see about us only. The T only. I'll select the canvas. See what we have done. First of all, sequences were generated, functional annotation, and classification. I have used this AgriGeo right now. But this AgriGeo, what are the results of this AgriGeo? We'll see in our results part. This is the gene ontology annotation that was done by using AgriGeo, and followed by that I use that annotate 8R. Where is the results? I'll see here. Here we have not mentioned this particular graph, no problem. I'll see my another publication where I have shown this graph. It's taking time, so I'll show you here only. This is the gene ontology annotation where I have done by using AgriGeo, and its model plant was basically rice. So what were the results? Yeah, these are the results. See, now we have identified how many genes which are related to replication, recombination, or other processes. And what is this A, B, and C? I'll open this in separate window so that it will be very clearly visible. Okay, this is basically, these are the particular pathways we have identified by using this. 1 minute. This is not. I think this figure is not visible in good form. So I'll show again Ciabotus only. Here it is. Now see this. But this is the microorganism. I have used Silo to Geo, I think, here. So we can easily find that how many genes are present in molecular functions, cellular components, and biological processes. And this is basically overall gene ontology annotation through NGS. You can open this publication and you can find gene ontology of some proteins in Ciabotus. This is molecular, this is biological, and this is cellular. In the same way, we have used in this particular organism, I have used Silo to Geo, but for plants, we have to use Agri to Geo. For humans, we can use Gorilla. So if you have any queries related to this ontology part, you can ask. Am I audible? Partha and Jash, do you have any query related to this ontology?